Aloha mai kako and welcome to a Botany 199 presentation. In this video, we review what we learned about energy in cells and the role of antioxidants in the health of a cell. Some of this material was originally presented by Dr. Lee during his first visit with us. Your homework that is associated with this presentation is to read a review article on the subject that Dr. Lee sent to us, and to write up a basic review of the subject that can be used as an introduction for your final write-up. The article and rubric for the writing will be posted to our Google Classroom and Blackboard page. Remember that you are made of trillions of individual units called cells. A typical animal cell is shown in the panel to the right. In ninth grade biology, you may have learned that each cell is like a small factory, with an administrative center called the nucleus that contains the DNA, and the ribosomes where machines called proteins are constructed. Recall that the powerhouse of the cell is the mitochondrion, the organelle where ATP is generated. Almost all cell types contain mitochondria, and a single human cell may contain hundreds to thousands of them depending on its individual energy requirements. Recall that adenosine triphosphate, or ATP, is the universal currency of energy in all living systems, including our own. The molecule is a modified adenine nucleotide with its three characteristic phosphate groups shown here in orange. Cells have hundreds of millions of ATP molecules within them to power all of their activities. In order to do any kind of work within a cell, ATP must be used. The way this works is that one of the phosphate groups on the ATP molecule is transferred to another molecule, a process called phosphorylation. This imparts energy to the system that the cell can use to do work. Here we see three examples. In the uppermost, energy from ATP is used to transport a molecule across a cell membrane. In the middle example, we can see how the phosphorylation of motor proteins leads to movement. This is how the actin and myosin filaments in your muscles are powered by ATP. Finally, at the bottom, we see how phosphorylation by ATP can be used to power chemical reactions in a cell. In this case, a synthesis reaction. Okay, let's start at the top of this diagram at 12 o'clock and work our way around clockwise. In this slide, the ATP molecule is shown as a blue ball with its three pink phosphate groups. Recall that a phosphate group is transferred to other molecules to provide energy for cells. This leaves us with the molecule of adenosine diphosphate, or ADP, which is missing one phosphate group. This is shown at the bottom of the diagram. In order to regenerate ATP from ADP, we must reattach the phosphate groups to the ADP molecules using energy from foods. This process occurs within the mitochondria of our cells. If you can remember back far enough, you'll recall that the process of regenerating ATP molecules from foods we eat is called cellular respiration. The overall reaction is shown below. Sugars from the foods we eat are chemically combined with oxygen that we get from breathing. The resulting products are carbon dioxide gas and water, which we exhale. The reaction is exothermic and releases energy that is used to regenerate ATP molecules. Cellular respiration includes a series of oxidation and reduction reactions, or redox reactions that you might remember from your chemistry class. Oxidation and reduction reactions occur together in pairs. One molecule loses electrons and becomes oxidized, and another gains those electrons to become reduced. Oxidation is losing electrons, and reduction is gaining them. This is where the terms oxidant and antioxidant come from, but more on that later. The redox reactions of cellular respiration occur in three distinct stages: glycolysis, which occurs outside the mitochondrion in the cytoplasm, and the Krebs cycle and electron transport chains that occur within the organelle. Each stage of cellular respiration includes several individual reactions, where electrons are eventually passed from one molecule to another through redox reactions, slowly releasing energy to regenerate ATP. Here we see the first 5 reactions in glycolysis. And here are the next 5 reactions. Cellular respiration works in this way, with several steps. This increases the efficiency of the processes and allows your cells to extract as much energy as possible from the foods you eat. Here are the collective reactions of the Krebs cycle, the second stage of cellular respiration. The details are not that important here, but but notice all of the individual reactions and intermediate molecules that are produced as energy is extracted. The final stage of cellular respiration is the electron transport chain of the inner mitochondrial membrane. In a final series of redox reactions, electrons are passed from carrier to carrier and eventually to oxygen, which liberates enough energy to regenerate many molecules of ATP. This is why we need to breathe, to supply oxygen to our cells so that we can use it to regenerate ATP from the foods we eat. All of this energy production is interrelated to all other metabolic pathways in your cells in an intricate series of thousands of individual chemical reactions summarized here. Okay, enough review. Finally, we get to our Botany 199 project, measuring antioxidant properties of extracts from plants. Free radicals, sometimes called reactive oxygen species or ROS, are forms of oxygen that are often created as byproducts of all the individual reactions that go into cellular respiration. Therefore, as our mitochondria regenerate ATP in our cells, these free radicals can build up and cause damage. Some of these reactive oxygen species are shown below. Superoxide anions, peroxides, and hydroxyl ions are all oxidants. They can build up during cellular respiration and remove electrons from other important molecules like proteins, lipids in the cell membrane, and DNA in the nucleus. Free radicals can also play an important role in cell signaling and defense. They play important roles in inflammatory and infection responses and can cause the death of a cell that is infected with a virus, for example. Reactive oxygen species can also be formed by exposure to environmental mutagens such as UV light or cigarette smoke. The action of free radicals causes a chain reaction of damage in cells. As reactive oxygen species remove electrons from important compounds, they also convert them into new free radicals that can in turn damage other parts of the cell. The buildup of these oxidant molecules in the cell is called oxidative stress. Oxidative stress has been linked to many chronic diseases, especially diseases of old age like heart disease, cancer, arthritis, Alzheimer's disease, and many others. They are the result of years and years of damage to cells by reactive oxygen species and the buildup of oxidants in your cells over time. As you might imagine, our cells have built-in ways to control the number of free radicals within our cells to maintain a proper balance. Remember, some free radicals are necessary because they play an important role in cell signaling and the control of the cell cycle, so a cell cannot neutralize all its free radicals. Enzymes like superoxidase dismutase, catalase, and glutathione peroxidase are all naturally produced within cells and have the function of neutralizing or scavenging excess free radicals that are produced by cells. Molecules such as these, which neutralize or scavenge free radicals, are called antioxidants. Other sources of antioxidants include the foods we eat, especially plants, that are filled with phytochemicals with the ability to neutralize free radicals. There are also a number of vitamins and other antioxidant supplements on the market. Here is a table that summarizes various sources of compounds that have been shown to have antioxidant properties in the lab. So how do antioxidants actually work to scavenge free radicals? Well, they're able to donate electrons to these reactive oxygen species, which inactivates them and stops the chain reaction of further free radical production. Most health-conscious people are aware of the anti-aging potential of antioxidant molecules, and that many kinds of antioxidants can be found in plants or acquired through supplements. The market for these molecules is ginormous, over 3 billion annually today, and projected to reach 4.5 billion in a couple of years. A brief tour of the supplement aisle at Costco will confirm this. In addition to vitamins like vitamins E, C, and A, which have shown to have remarkable antioxidant properties in the lab, there are many plant-based chemicals called phytochemicals that exhibit similar antioxidant properties. The phytochemicals that exhibit the greatest antioxidant activity in the lab are polyphenols. Polyphenols are a class of compounds that come in a wide variety of forms and are relatively abundant in many plants. Polyphenols play a part in a plant's secondary chemistry,
those chemical pathways that are unrelated to photosynthesis. In plants, polyphenolic compounds play important roles in growth regulation and structure.
Flavonoids are the most abundant class of polyphenolic compounds. They are rich in hydroxyl or OH groups and are very effective at neutralizing free radicals in cells. They also have the added benefit of chelating, or binding to metal ions in the cell and preventing them from causing more cellular damage. Please revisit our class notes on secondary chemistry of plants for a review of these molecules. This slide highlights some examples of flavonoids with antioxidant properties. For example, anthocyanins, those molecules that give color to blueberries, blackberries, and cranberries. Or catechins found in chocolate and green tea. Or resveratrol, an important adult justification for another glass of red wine. Notice that all of these molecules have similar structures and are rich in OH groups. Here is a list and some pictures of foods that are rich in antioxidant phytochemicals. Notice that many of them are colorful and aromatic. We can measure the ability of a molecule to neutralize reactive oxygen species in the lab with an assay called Oxygen Radical Absorbance Capacity, or ORAC. It is important to point out that just because a molecule is shown to have antioxidant properties in a test tube, this does not mean that it acts as an antioxidant when it is taken in as food. We have been assessing the ability of our plant extracts to neutralize free radicals in the lab, but this does not necessarily mean that the extracts from our plants will act as antioxidants after being absorbed by digestion. Wait, what? I sat through 20 minutes to hear that? Well, just because a molecule does not act as an antioxidant in cells, it does not mean that it doesn't have positive effects. A molecule may help to regulate naturally occurring antioxidants in cells, or may act to regulate cell signaling in other ways. Many phytochemicals have been shown to have other remarkable effects in both laboratory and clinical trials as listed here. Here are some examples of how this works. Flavonoid molecules that act as antioxidants in the lab may actually provide health benefits by modulating the inflammatory response, slowing the oxidation of low-density lipoproteins, or inhibiting platelet aggregations in cells. Despite our lack of knowledge, the antioxidant supplement market continues to grow. We still do not know the optimal levels of intake for these compounds, and excessive amounts are excreted or can cause damage. There is still much to be learned, and this learning happens in small steps like the experiments we conducted at WCC earlier this year. So what's next? First, read the review article called Antioxidants: Characterization, Natural Sources, and Extraction and Analysis provided by Dr. Lee and posted to our Google Classroom. Use your notes from the reading, this presentation, as well as your own internet research to summarize the reasoning behind our research this semester. See our Google Classroom for a rubric and timeline for this assignment. Please stay on top of this. I want to address our data next week and use this momentum to finish off our Botany 199 project within the next few weeks. The next slide has some important references on it that you might be interested in. Thanks for listening.